Thank you all for the opportunity to speak at this workshop. I really hope my presentation today will really provide you with a glimpse of the various research questions and approaches that we could address using FTICR datasets. So as you all know, climate change is anticipated to induce a multitude of transformations on Earth. These alterations include rising temperatures, as well as intensified and prolonged drought episodes, and erratic precipitation patterns. These environmental shifts are projected to significantly impact both plant and microbial activities, which in return will impact ecosystem processes and function. Now, tracking organic compounds through metabolomics or organic carbon characterization is really crucial for assessing the impact of climate change on ecosystem function, because it offers insights into carbon cycling and ecosystem resilience. It also extends beyond genomics to reveal metabolic responses, phenotypic plasticity, and broader biogeochemical processes, therefore aiding in our understanding of climate-induced environmental changes. So now I want to go over to talk a little bit about how we're going to be using FTICR data to understand or track changes in biological processes with changing environmental conditions. So Will already showed you an example of the stoichiometric analysis that can be used with FTICR-MS. Here I'm going to be focusing on additional type of analysis that we could perform with this rich data set. Keep in mind that these data sets are really versatile and can be used in so many different ways, depending on the specific research questions you are addressing. For example, you could utilize FTICR data at the feature level, that is looking at the features regardless of whether they are assigned a molecular formula or not, to compute various indices that provide insights into the changes in organic matter richness or diversity as a function of treatment, depth, location, and so on. Using the same data set, you could also perform a wide range of statistical analysis. Like Will mentioned, these include NMDS plots, PermaNova, to really assess the similarity or dissimilarity of organic compound compositions across multiple samples under different treatments, depth, locations, and so on. Additionally, you could classify each feature based on its molecular formula to distinguish between nitrogen-containing compounds, phosphorus-containing compounds, and so on, as well as to distinguish between the different biogeochemical classes of compounds, such as lignin-like compounds, sugar-like compounds, and so on. Then you could use the relative abundance of these elemental compositions for PCA analysis to examine, for example, how the lignin-like composition or abundance is changing between samples, between depth, and treatments. Another approach to visualize all the compounds that received a chemical formula is using different plots. Some of them Will mention in his talk. These include Van Krevelen plots. You could also do oxygen distribution plots, carbon plots, and so on. Additionally, you could take the molecular formula and map them into KEGG, which is a Kyoto Encyclopedia of Genes database, to give you an idea about the potential pathways that these metabolites are involved in or produced by. As Will mention, these are molecular formulas only, and each molecular formula could be multiple compounds. So that's why when you do the mapping to KEGG, you have to take that with a grain of salt, because here you're only getting an idea about the potential pathways. For example, I could map my metabolite into KEGG, and I could see that these metabolites are involved in biosynthesis of amino acids or glycolysis or so. Now, since these are only potential matches, you then have to validate these data, either using metatranscriptomics analysis or liquid chromatography mass spec, where you could have more accurate annotation of molecular compound. But regardless of that, FTICR data could be used to just give you an idea about the potential pathways that the microbial community are doing in your ecosystem. Now, another approach is to visualize another approach that also takes into consideration all the features observed in a mass spectrum is using transformation analysis. Here, you could use the same data set to look at differences in reactions responsible for the production or transformation of organic compounds. This approach is distinct from pathways because it specifies exact reactions. For example, I am looking at a mass loss of hydrogen, a mass loss of nitrogen, or methyl, and so on. These could occur in multiple pathways. But again, regardless of which approach you want to use, it at the end of the day depends on the research question you are asking. But again, as I mentioned earlier, this is a really rich data set and you could do a lot with it. And we're always looking forward to new ways by which people could use these data to answer their scientific questions. Now, I've been fortunate to apply these approaches to answer many questions across multiple disciplines. For this talk today, I'll be mostly focusing on the environmental field, differentiating between organism level analysis and ecosystem level analysis. In particular, today, I'll be focusing on a couple projects only to give you an idea about how FTICR allowed us to address our research questions at the broad ecosystem level. Now, the initial study I like to emphasize is our research in the Sonoran Desert. I live in the desert, so it makes sense to do research in the Sonoran Desert. So my team and I have been monitoring the system since 2021. Our objective here was to understand the impact of monsoon rain on microbial activity. This is really important to us because we anticipate that this system, as well as many other systems in southwestern USA, will become even hotter with increased episodes of heavy rain with climate change. Therefore, it's really important for us to assess at the moment how resilient these ecosystems. This will then enable us to set a reference point for comparisons with evolving environmental conditions. So to do this study, what we've done, we gathered soil samples across the different stages of the monsoon season. If you look at the figure on the bottom, you could see variations in the soil moisture across these three monsoon stages. For example, in the pre-monsoon season, we had 2 sampling events from May and June. Then during the monsoon season, which is usually between July and August, we were able to collect samples from 4 events where we had oscillating dry, wet events. And then at the end of the monsoon season, we went ahead and collected samples from September and October. And as you could see, the soil moisture is starting to decrease to go back to the starting conditions. We then conducted a comprehensive analysis of these samples, employing a wide range of techniques, every single possible technique, but also including FTIC-RMS, with, again, the main goal here is to monitor alterations in biogeochemical processes throughout the 3 monsoon stages. For the talk today, I'll be showing you some of the microbial data, but mostly focusing on the FTIC-RMS data. So I'm going to start first with quickly with the 16S data, where we are looking at the composition of the microbial communities across the monsoon season. So when initially investigating alterations in the composition of the microbial community, it was surprising to us to find no significant changes in the composition of the microbial communities that lives in the Sonoran Desert across the 3 stages of the monsoon. I'll be presenting the results in 2 distinct formats. First, I have an NMDS plot here shown on the left, illustrating the absence of significant changes in the composition of the microbial community, suggesting at the moment that the composition of the microbial community is really stable and therefore does not really care whether there is monsoon rain or there is no monsoon rain. Another way to visualize the data is through the use of a bar diagram shown here on the right, where I am looking at the change in the relative abundance of the different classes of bacteria that lives in the Sonoran Desert across the 3 monsoon season. Again, you could see that there is not much significant changes in the abundance of the microbial communities throughout the monsoon season. Another intriguing observation is that the bacterial composition was predominantly characterized by thermoeulophilia. These are thermophilic organisms, as well as actinobacteria here in actinobacteria in lighter blue. And these organisms are known for their resistance to high temperature and desiccation. So these findings at the moment suggest that bacterial composition displays resilience or resistance, which could be facilitated by their ability to change what they are doing depending on the environmental conditions and the substrate available. So the take-home message at this moment is microbial composition is really stable. But we know that the microbes, to be able to live in the desert, they should be able to change what they are doing depending on the changing environmental conditions. So to kind of go back and validate these hypotheses, we then used both metatranscriptomics data and metabolomics data to see the function of the microbial communities across the 3 monsoon stages. Here, I'll be focusing mostly on the organic carbon characterization data. I'm presenting the data here on the left in the form of an ordination analysis. Here, we're using a supervised PLSDA analysis, where each dot represents a sample collected from different months, which corresponds to different monsoon stage. And what I'm gonna do, I'm gonna show you how the composition of organic matter is changing as a function of the monsoon season. You can see as the monsoon season starts, this is where the pre-monsoon season is, where the red circle is. And then as the monsoon season starts, we see a significant shift in the composition of organic carbon. Again, remember that most of the carbon we see in the desert soil is due to the microbial activity. So these data so far suggest confirm our hypothesis that even though the composition of the microbial community is stable, what they're doing is actually changing. So where we have a compositional stability, we have a change in the functional profile in the microbial communities. As the functional activity of the microbial communities changes, then we observe that in our organic carbon characterization data. But what's really cool is that at the end of the monsoon season, you see that the organic carbon composition goes back to the starting conditions. These suggest that microbial communities in the Sonoran Desert are not only resistant, that is, they keep their composition fixed throughout the year, but also they are resilient. They are able to go back to where they started each and every single year, which was really cool for us to see and was really useful to use FTICR data to allow us to confirm our hypothesis that the functional activity of the microbes is changing. Microbial communities in the desert have adapted to these changing environmental conditions by keeping their composition stable, but changing what they are doing. What we've done next was we take those metabolite data and map them onto KEGG to identify the different pathways by which the microbial communities are overcoming the change in the soil moisture. I'm showing the data here on the right, and I will walk you through this a little bit complicated figure. But what we have here is we focused on the top 10, 15 pathways that appear to be active in the Sonoran Desert soil. And then we wanted to see how many metabolites mapped each pathway and compared between different months. For example, if we look at the first column, we're looking at May versus June. Both of these months are in the pre-monsoon phase. And you can see we do not have significant changes in the number of metabolites that mapped each pathway before the monsoon season. But now if I take May and compare that to July 1st, or the first sampling event in July, which is the second column, you can see now we have a significant increase in the number of metabolites that map each of these pathways, consistent with our hypothesis that microbial activity is increasing with the monsoon season. And apparently, as the monsoon season comes, the microbial communities now start turning on and off specific gene to carry out specific reactions. For example, we see an increase in the biosynthesis of amino acids, ABC transporters, mineral absorption. Having the ability to change what they're doing is what's allowing the microbial communities to continue to live in the Sonoran Desert. And you could see that if we take any May and compare it to any of these 4 sampling events in the monsoon season, we continue to see evidence of microbial activity. However, at the end of the monsoon season, if we now take May and compare it to September, or May and compared it to October, we see that the number of metabolites continue to, the difference between May and September start to decrease again, consistent with the fact that at the end of the monsoon season, the microbial communities go back to their starting condition. So this is a quick example of how we actually use FTICR data to understand how the monsoon season influenced biogeochemical cycling in the Sonoran Desert. We now continue to monitor the site for the coming few years to see if there is a threshold above which the microbial communities are not resistant or resilient anymore. Now, another study I like to highlight took place at the Spruce Experimental Forest in northern Minnesota. That was in collaboration with Natalie Griffins at Oak Ridge National Lab. Sorry, I thought
somebody was coughing. So
all right, sorry. All right. So the primary objective of this study was to investigate how different types of litter decompose under varying temperature conditions. For those of you who are not familiar with the Spruce experiment, this is funded by DOE, where they build these huge chambers to understand how a peat bog will be influenced by elevated temperature and CO2. And you can see on the figure on the left, we have different chambers, and each chamber received a different treatment, either elevated CO2, elevated temperature, or just ambient CO2 versus high temperature, and so on. Now, what we've done here, we decided to use 3 different plant litter: sphagnum moss, Labrador tea, and spruce. And we put those in mesh bags, and these mesh bags were then put inside the chambers. And here we wanted to understand, do plant species decompose differently based on the type of the plant? But also, how would elevated temperature and CO2 influence how organic matter gets decomposed? So now I'm going to show you how we use FTICR MS data to help us understand, do organic matter decompose the same way based on the temperature, CO2 treatment, or the plant species type is what dictates the change in the organic matter composition with time. Here I'm showing the data as an NMDS plot. Here is the NMDS plot, where I have the different colors represent different plant species. For example, pink and purple are sphagnum. Then we have the green are the spruce, leaves and root. And then we have the lab tea, it's kind of orangey and yellow. And the different shapes represent the different pickup time. Here, I'm not coloring or differentiating between the different chambers. For the reason is, if you look at this NMDS plot, you clearly see that the major driver in the change in the organic matter composition, it's first the type of plant. For example, we can see that the sphagnum here in purple and pink is really different than the rest of the plants, but also you can see the effect of the time. And as you can see in our subsequent analysis, it turned out what dictates the changes in the organic matter composition is mostly the plant type, as well as the pickup time. We picked the samples across 2 years, where we went to the field and collected those mesh bags across 4 time events within the 2 years. And then we saw a little effect of the temperature and the CO2 to our surprise. Now, to further validate these results, we did something different. So what we've done, we took these data from a feature level, and then we calculated the relative abundance of the different classes of compounds, such as amino, sugar, carbohydrates, lignins, the abundance of CHO compounds, CHNO. We also calculated some of these stoichiometric measurements that we'll talk about, double bond equivalent, Gibbs free energy aromaticity index. And then we employed something called CCA analysis, or canonical correspondence analysis, to figure out what is the driver in the change in the organic matter composition across all these samples. Because remember, we have different litter type, we have different pickup type, we have different temperature treatment, different CO2 treatment. So we wanted one way to help us figure out what is driving these differences. So our results are shown here on the bottom right, where you can clearly see the bigger the circle, the more significant that variable is. We clearly see that the litter type is really significant driver of the change in the organic matter composition, followed by pickup time. We also saw some significance of the interaction between temperature and litter, But as you could see that the temperature and the CO2 were less driver of the change in the organic matter composition compared to the litter type and the pickup type. So now we use CCA analysis to tell us how we should move forward with looking at the data. Now what I'm going to show you is how we took one plant species to figure out how organic matter is decomposing, because we know that plant species type is the major driver of changes in the organic matter composition. So I'm going to be focusing mostly on sphagnum. This is a principal component analysis based on the abundance of the different classes of compounds as a function of time. This plot clearly illustrates how organic matter composition is changing of time. What is really cool is you kind of see a clockwise distribution or a change in the organic matter from T0, where we have a lot of peptides, some lipids, amino sugars. And then as the time goes by, we start seeing an increase in the lignin. And at the end of 2 years, we have an increase in the abundance of condensed aromatic and tannin. So what is this suggesting is that the microbial communities initially start consuming easily degradable compounds, leaving behind hard to decompose substance. Now we could represent this data differently using Gibbs-free energy violin plots. Again, for those that are not familiar with the Gibbs-free energy analysis, the higher the value, the more easier the compound to be decomposed, or the organisms will get more energy from it. As the values go down, this compound becomes harder to decompose, or the organisms will not gain much energy from it. And as you could see, as we move from T0 to T2, which is after 2 years of leaving these mesh bags in the enclosures, we see a significant decrease in the Gibbs-free energy, consistent with what we observed in the PCA plot. As we move from T0 to T2, we are consuming all these labile, yummy compounds such as peptides, amino sugars, and then leaving behind those hard to decompose organic compounds such as lignins, tannins, and condensed aromatics. Lastly, we could use the same data set to calculate a different index called metabolite diversity, where we could see an significant increase after half a year of putting these bags in the enclosures, where literal decomposition reaches a maximum, followed by a decrease in T1 and T2. However, it continued to be above the baseline. This pattern is really common in decomposition studies, as the breakdown of organic compounds lead to increased diversity due to the metabolic transformation of organic matter. So this is again another quick example of the different ways by which you could use the same data set, but also try to get different ideas of what's going on in your assignment. I don't have time to go over all the different analysis, but I want to highlight 3 additional analyses. You could also use your FTICR data to generate some metabolite microbe interaction networks, and then track how these microbe metabolite interaction networks is changing with changing environmental conditions. Here I'm showing how these networks are changing with thaw. You could also use some assembly processes, an approach developed by James Teagan's group, where you could look at what is driving the change in organic matter composition, whether it's stochastic or deterministic. And finally, you could use some transformation analysis to really allow you to understand the different reactions by which organic matter is being transformed or degraded into your system. So I really hope this gave you a glimpse of how we can use the data. As I mentioned, there's so many different ways that we don't have time to talk about. I really encourage you to really be creative in using these data sets. These are really rich, versatile data sets. And as I mentioned earlier, we're really looking forward of new ways of using this data beyond what is currently available in the literature. And I know that you're going to have some additional tutorials this afternoon to show you some of the different ways you could visualize these data. But as I mentioned, I encourage you to also think about new ways you could use these data. With that, thank you so much for giving me this opportunity. And sorry I couldn't be there with you in person. Thank you. I'll take any questions if there is any.
Thank you, Malak. This is fantastic talk. There is a question in the Zoom. I can read it. This is from Avni Malhotra. Yeah, so super nice talk. She says, do you want to answer it or do you want me to?
Yeah, I'm reading. So do you also interpret the variance explained by definition? Yeah. So as you mentioned, if you look at the variance, we have 15% roughly due to the monsoon season, and then we have kind of 20% due to the variation in the sites. So there's a lot of things happens during the monsoon season, and I agree that 15% is not too much. But again, because we have, as you mentioned, when the monsoon season comes in, we have so much going on. The monsoon season comes so quick and fast. We have so much movement of the substrate around. So we have a lot of variation due to the sampling location. So that's why we ended up having to use a supervised method to be able to capture some of these differences. And then we validated these using transcriptomics analysis, because the sites, we had so much heterogeneity between the sites, again, depending on the flow of water in the months in the desert. So that's why 15% and 20% are not necessarily too much, but considering the complexity of the system and the heterogeneity of the soil samples, this was enough for us to take these observations and validate with transcriptomics data. But that's, again, an example of why we had to use a supervised method, because when we did NMDS analysis, we could not see much changes. It was only with supervised method we were able to capture some of these changes, but we had an orthogonal measurement to validate these observations. I hope this answered your question. Thank you.
Thank you. There is also Sutapa Gosal. He has a question, has raised his hand. Can you go ahead, unmute? Yeah.
Okay. Hi there. It was a wonderful presentation.
Thank you.
I'm new to this area, so I had a question regarding the initial thing that you showed about in the you did over the year measurements, pre-monsoon, monsoon, and then post-monsoon, right?
Yes.
And you observed that the microbial community is able to survive during the whole annual season.
Yeah.
So that is somewhat similar to like in the animal world also, like we do different things and we are able to survive because we are aware of what the changes are going to be over the years. So what I was wondering that, but if the time of the monsoon, the amount of monsoon that you receive decreases significantly, then that cycle is interrupted and the organisms are going to react in a way. So is it possible that you measure the regular time period over a season, and then when that time thing is not regular, then you do measurements again and you can see how the organisms react in a different sequence of events?
That's exactly true. And that's why we are actually tracking the system for at least 5 years to be able to see, because we started in 2021. And again, as you mentioned, it could be that an off season or not. So that's why we're going to be tracking it for multiple years to better understand really how the system is, if it's oscillating, because each year, if you live here in the desert, each year is completely different. So we started in 2021 and we have collected samples in 2022, 2023, and we are doing the same analysis up to 2025. And it's exactly for that particular reason. We want to see how these behaviors is changing, especially that each year we are seeing differences in the monsoon season. In 2021, we had a beautiful monsoon season. We have lots of rain. Then in 2022, we had less rain. In 2023, we had less rain. So now that's why we're going to continue tracking the system for up to 5 years to be able to see the changes from one year to another, so that we could have a better idea of how resilient that system at the moment. But within the first year, it looked really good. But again, we have to look at 2022, 2023, 24, and 25 to see if each year is different depending on the monsoon season.
Yeah, that's exactly means, in terms of figuring out how the environment is going to react, one first has to establish a normal reaction, then be able to distinguish it from that normalcy as the environment changes.
Exactly. Yeah.
Okay. Thank you very much.
Thank you.